So this video will be an outline of quality control for sequencing. And I want to start by going over why we even bother to quality control. So what you see on the screen here is on the left an Illumina flow cell. So this is the actual physical thing that we sequence from. And so as you've probably learned earlier, on the flow cell we have clusters, and each cluster is basically copies of some library component that we're going to sequence, and they're all identical, right? And so each cycle of sequencing produces an image like this for each of the fluorescent channels, A, G, C, and T. And so we just see this. And so for each cluster, we see just a dot. And that's really all that sequencing is. I think that's the key point to take away here, is that Illumina sequencing is just really a camera staring at a bunch of flashing lights. And so there are a lot of ways that you can get error with that, as you might imagine. So in the initial few cycles of analysis of this process, you actually have to define where are the clusters, define the clusters. And if there's not a lot of diversity in the sequence initially, or if we over-under cluster, so they're either too dense or they're too far apart, we can have issues that come from that. And there's a lot of things that come up from the fact this really is a physical process. And so a lot of the issues with quality control, with error, can be understood just by thinking back to what is physically happening during this step of the sequencing process. And so what are some sources of error? So we have sample quality. So the input of your DNA, your RNA, is it fragmented? Is there enough of it? All these factors can create error. So fragmented sample will create generally poor quality libraries, and that can be visible in a number of ways. And of course, if you have not enough input, that can create all sorts of issues as well, primer dimers and so on. And there's library prep issues. So every single step in your library prep has bias associated with it. So I guess error or bias. So you can either mess up and cause something bad to happen, or just there's an inherent bias to every single step. So a polymerase is not have equal affinity to every step in a reaction, to every possible substrate, right? So some nucleic acid substrates will be more favorable, some will be less favorable. And so there's always, every single manipulation you do biases your output. And that's something to keep in mind at all times, is that you can't really get away from this. You can minimize it, you can account for it, but no matter what you do, every manipulation, every step will in some way change your sample, bias your sample. Then there's the issue of contamination. So this is could be a lot easier than you think. So the issue with sequencing is that we can detect very, very, very low levels of contamination in our library. And this can come from something as simple as, if I'm preparing my library at a bench across the lab from you, and you're preparing your library, and I'm working with mice DNA, you're working with rat DNA, just the aerosol production from your pipetting can be enough to introduce detectable contamination to my sample. And this really isn't an issue for a lot of things, right? You can just filter it out, or just ignore it. But for certain things like pathogen detection or things like that, microbiome studies, contamination is a huge issue, right? Anything where you care about low abundance things, this can be a big problem. And so you need to be able to account for it and detect it. And there's a lot of software out there that will let you map to multiple different genomes that are possible sources of contamination. So generally, it's not a bad idea to screen for anything that you work with to see if it's there. And of course, then there's sequencing error, like I talked about earlier, under-clustering, over-clustering, low diversity, and so on. So one general principle is that quality will decay over a read. So I'm going to have higher quality at the start of a read and lower quality at the end. And this really becomes amplified the longer a read is. So just something to keep in mind there is that you want to have that drop-off in quality be acceptable. And this would be an example of an unacceptable drop in quality. So what are situations where you should really be particularly concerned about quality control? Something that you really have to keep an eye on. So high or low GC content. This comes back to the idea that enzymes and other steps in the process have different affinities. Well, they don't deal that well with unusual GC content, either regions or genomes. So if you're looking at regions that have high or low GC content, or you're looking at a genome that has unusual GC content, you should really watch out for those kinds of issues. Basically, sequencing protocols are generally optimized to work with standard GC content organisms like human, like E. coli. And if you're well away from that, there are going to be some problems they have to look out for. There are paired end differences. So it's known that you can have situations fairly frequently where the second read in a pair will have lower quality than the first read. That's fine as long as it doesn't get out of hand, so it's something to keep an eye on. Particularly long reads. If you're doing, 2 by 250 base pair sequencing, 2 by 300, whatever it is, you're going to have a lot more issue with quality than you are with 2 by 50 base pair sequence. And so you need to keep an eye on that. Just see how quick is my quality drop off, and am I really gaining anything, from going long? And that's going to depend on a lot of different factors. And there's low diversity sequencing. So for example, amplicons. So if I'm sequencing 16S ribosomal RNA, or ribosomal DNA for that matter, the initial bases in every single sample are going to be quite similar, right? And that can be a huge problem because it's going to be hard for the processing to work correctly to separate out clusters from each other and detect them. And this also goes for homopolymer regions where I have the same base over and over again. It can be very easy to mistake that. And so with low diversity sequencing, there are a lot of ways to go about this, ways to introduce diversity. Using barcodes, for example, can help you get around some issues. Spiking in other sequence that I don't care about, but that provides diversity. Phi-X is often used for this. And so just if you're doing something like this, one of these kind of special cases, you're going to need to do some extra reading and extra steps to make sure that you're doing things right. And the reason we really care about quality is that even if we don't have huge blatant error, quality really does influence downstream steps. So the higher quality the read is, the more confidence we have in each base call, the more unique it is, the more mappable it is to that genome. And essentially everything we do, variant calling, de novo assembly, transcriptomics, RNA-seq, all of this depends on mapping. At some point, you're probably going to have to map your reads to a genome. And the higher quality your reads are, the better you're able to do that. And of course, the reason we have coverage, the reason we talk about coverage is that we know there's always going to be error. There's always going to be some rate of error. And if we have 30x coverage, kind of the standard for genome resequencing, then we know that we can compensate for those errors just by looking within our reads. And so we need a way to quantify base quality. We need a way to say how good is our base call at this point. And the way we do that with Phred scores. So we have some probability P of an incorrect base call. So the probability that this base, A, G, C, T, whatever it is not actually that, but is something else. And so this is going to be very dependent, the way it's calculated, on the platform you're using, on the machine you're using. But somehow that manufacturer's on an algorithm that looks at it and says, okay, here's my confidence that I'm going to pull out. And so we take log 10 of that probability, we multiply it by -10, and we get out this value Q, which is a Phred quality score. And so the higher that score is, the higher quality we are, and the lower the probability of an incorrect call at that base. And so one thing you need to look out for is nucleotide distribution. So you want to see that your GC content in your reads basically fits your GC content in your genome. And so we kind of expect that the GC content in a perfect scenario will follow normal distribution with a mean of the mean GC content in your entire genome. And so here we have a case where in red, the actual experimental distribution in our reads doesn't match up with that theoretical distribution. So that indicates that something went wrong here, that somehow we were favoring certain regions in the genome over others perhaps. So it's just something to watch out for there. And then we have library prep biases. So again, not all inputs are created equal. Different steps have different selectivity for different nucleic acid patterns, basically. And GC content affects library prep efficiencies, and every step is biased. So random hexamers are not actually random. They favor certain parts of the genome. Ligation, not actually random, will favor certain substrates. And so no matter what we do, we introduce bias. And we have to remember that and basically minimize our manipulations as much as possible. There's also adapter and primer contamination. So this is cases where our adapter sequences and our primer sequences from the sample prep get sequenced. So primer dimers are the primary way that we get primer sequences. So if we have a poor quality or low concentration input, and if our primers have some affinity for each other, they'll bind to each other, form a double strand, be extended by the polymerase, and then become introduced into our library potentially. And so this is something that you watch out for and screen for. Adapter contamination can happen, for example, if our sequencing primer is binding upstream where it should be, or if our insert is too short. Issues like that can lead to a sequencing adapter, and of course we'll want to remove that from our sequencing. And how do we look for these things? We have what's called read trimming quality control programs. There really are a lot of ways to do this, a lot of software. It's also something that can be kind of interesting to implement yourself, but really just I want to provide some names here so that you know where to look when you're doing this. And so Sickle and Trim Galore are both good options, where they'll do both quality-based trimming, where they say, okay, we'll trim from the ends with some kind of quality threshold to how far we trim to get away low quality regions at the each end of the read. And they'll also look for homology with adapter and primer sequences that are either predefined based on, sort of what are the standard Illumina adapters, or that you can define yourself and look for that and say, okay, if we see that in the read at the beginning or end, we'll trim it away. And so that's a very important quality control step. And another very important part of quality control is just looking at all these metrics like quality of a read, GC content, and so on. And the software we're going to use to look at that is called FastQC. So there's a tutorial video here at this link that you should look at, and I think that will be on the course website. And then there's also command line use. So that's useful if we're putting together a pipeline, a script, we can implement it from the command line. So you just /FastQC --help gives you all the options. And to run with all the default settings, just /FastQC, whatever your FastQ file is. You can also run it with SAM or BAM files. And then we have the report, which will be in HTML. And to look at that, you just run Firefox and the report.html. The key point here is that if you're doing this on a server, you have to have connected with X forwarding enabled. So that lets you basically look at GUI graphical software on the server on your own screen. And so the way you do that is SSH -x, put in the -x, your username at the server .colorado.edu.